Hello everyone and welcome to our channel Mootechie. We're so glad to have you here. Today we're diving deep into the fascinating world of milk, exploring its physical chemistry, from the basic composition to the complex structures that make it such a unique and vital substance. Let's get started. Now, let's begin by looking at the various factors that affect what milk is made of. First up, we have the animal factor. This includes genetics, the specific species and breed, the individual cow itself, and the stage of its lactation period. Then we have age and genetic factors. Breeds encompass breed, species, feed, and the cow's individuality. The stage of lactation is crucial, along with pregnancy and the nutritional balance of the animal's diet. We also must consider the health status of the cow, including events like oestrus and gestation, and importantly, the presence of any mastitic infection. And finally, environmental factors play a role, such as extreme climates, stress, exhaustion, and even the type of housing. Milking technique, milking frequency, and the stage of milking also have a significant impact. Now let's move on to a fascinating comparison of milk from different species. This table shows the composition in percentage of milk from various dairying species. For human milk, total solids are 12.2%, fat is 3.8%, protein is 1.0%, lactose is 7.0%, and ash is 0.2%. For cow milk, total solids are 12.7%, fat is 3.7%, protein is 3.4%, lactose is 4.8%, and ash is 0.7%. So on. And finally, for the grey seal, total solids are an astounding 67.7%, fat is 53.1%, protein is 11.2%, lactose is 0.7%, and ash is not specified. It's amazing how different milk can be across the animal kingdom. Coming up, we explore the most fundamental component of milk, water. Water is the principal constituent in milk, making up 79% to 90% of it depending on the species. It encompasses all other constituents of milk, which we call the total solids, that are either dissolved or suspended in it. Small amounts of water are hydrated or bound chemically to lactose, salt, or protein. The water activity in milk is relatively high, at 0.993. Removal of water increases shelf life, which is the principle behind making powdered milk. And importantly, regulations prohibit the addition of water to raw milk to prevent adulteration. Next, we delve into the world of milk fat. Milk fat is found in tiny spheres called fat globules, which are protected by a membrane, known as the fat globule membrane, or FGM. These fat globules range from 1 to 20 micrometers in diameter. They are made up of approximately 98% triglycerides, 0.2% to 1% phospholipids, and 0.2% to 0.4% sterols. The phospholipids and proteins mostly associate with the fat globule membrane, which is key to its stability. Let's understand milk fat a little more deeply. It contains traces of fatty acids, vitamins A, D, E, and K, and even some enzymes. Incredibly, there are more than 400 different fatty acids identified in milk. The predominant fatty acids in bovine milk are myristic acid, also known as C14, is to 0, palmitic acid, AID, or C16, is to 0, stearic acid, C18, is to 0, and oleic acid, C18, is to 1. Now, let's visualize the structure of a milk fat globule. This diagram shows a natural milk fat globule. At its core are the triglycerides. It's surrounded by a primary membrane, which is a double layer derived from the plasmic membrane of the cell, and it includes something called a cytoplasmic crescent. Then we have the detailed milk fat globule membrane, or MFGM. This contains high melting point triglycerides, low HLB phospholipids, an enzyme called xanthine oxidase, and high HLB phospholipids. The diagram also points to a glycosylated polypeptide on the surface. We see labels like XO for xanthine oxidase, and sizes like A, 10 micrometers, B, 1.5 micrometers, and C and D at 0.5 micrometers. Another component is a protein called butyrophilin. We also see cholesterol, glycolipids, and an enzyme called 5' nucleotidase. Now, the diagram also shows a mechanically disrupted milk fat globule DORH, which is smaller than 4 micrometers. This results in a native MFGM fragment, the exposure of serum proteins, and a smaller size labeled N, 180 nanometers. We also see a spread casein micelle subunit and a full casein micelle nearby, showing how these components interact. So why is this fat globule membrane so important? The lipids, lipoproteins, cerebrosides, nucleic acids, enzymes, trace elements, and some bound water molecules all help stabilize and prevent the fat globules from coalescing or joining together during milk processing and handling. The FGM prevents attack from lipases, which would cause lipolysis. Lipolysis would lead to an increase in the amount of diglycerides, monoglycerides, and free fatty acids in milk. These free fatty acids are fairly water-soluble and are situated in both the milk plasma and the fat itself. Let's continue with the properties of milk fat. The short free fatty acids situated in the milk plasma are ionized and more water-soluble than the long free fatty acids, those with more than 14 carbon atoms, which are found in the fat and at the oil-water interface. Minerals associated with the fat globule membrane include copper worry, which makes up 5 to 25% of the total, and iron guri, which constitutes 30 to 60%. Other minerals found here include cobalt, calcium, sodium, potassium, magnesium, manganese, molybdenum, and zinc. Furthermore, compound lipids also occur in milk, such as phospholipids and phosphatides. These are situated mainly in the fat globule membranes but also in the milk plasma, lipoproteins, and milk microsomes. Phospholipids and phosphatides are highly surface active and polar, and they dissolve poorly in both water and oil. It's also interesting to note that lipids can be crystallized, which affects the fat structure, its melting range, and the rheological properties or flow of the milk. There's more to learn about milk fat. Furthermore, autoxidation of the double fatty acid bonds or residues can occur, leading to off flavors, which is a major concern for dairy quality. Whole milk contains 10 to 20 mg per 100 grams of cholesterol in milk with 3.3% fat. The amount of cholesterol is positively correlated with the amount of fat in the product. Cholesterol is located in the fat globule membrane, and approximately 10% of the cholesterol is esterified. Now let's shift our focus to another major component, milk proteins. Proteins are made up of amino acids with specific properties that are determined by the side chains of the amino acids in the polypeptide chain. The conformation or shape of the protein depends on the hydrogen bonds, hydrophobic interactions, and salt bridges formed between the peptide chains. Regular arrangements within the protein include structures known as beta sheets and alpha helices. The structure of these proteins is not static. Temperature, ionic strength, and pH all affect protein conformation. The major classes of milk proteins are casein and whey, or serum proteins. These proteins are synthesized in the mammary gland, and their structure is derived genetically. The protein content of milk remains fairly constant, with a concentration range of 30 to 35 grams per kilogram. However, it is influenced by the lactation stage of the cow. Let's now zoom in on the casein proteins. There are 4 main types of casein, and they have genetic variants: alpha S1 casein, alpha S2 casein, beta casein, and kappa casein. They are phosphorylated and hydrophobic, and they associate with themselves and each other. They represent 38%, 10%, 36%, and 13% of whole casein respectively. Overall, an incredible 21 variants of casein have been identified that occur by genetic mutations. Caseins have some unique properties. They have distinct disordered molecular structures that lack disulfide bridges. They are very heat stable, withstanding temperatures above 140 degrees Celsius before dissociating. Their hydrophobicity is due to the high ratios of apolar amino acids, including valine, leucine, isoleucine, phenylalanine, and proline, which make up between 35% and 45% of their structure. This hydrophobicity is counteracted by the high phosphate content and the low concentrations of sulfur-containing amino acids such as methionine and cysteine. This balance allows the caseins to be reasonably water-soluble. Their susceptibility to proteolysis, or being broken down by enzymes, is due to the lack of secondary and tertiary structures, particularly in alpha S1 casein and beta casein. Now let's look at the alpha caseins in more detail. First, alpha-1 casein has 199 amino acids and is approximately 23.6 kilodaltons. It has the highest charge of all the casein molecules and consists of at least 8 phosphoserine units. It has 17 proline residues that ultimately disrupt the formation of secondary structures, such as alpha, helices, and beta sheets. Next, alpha-2 casein has 207 amino acids and is approximately 25.4 kilodaltons. It is the least hydrophobic casein molecule. It has several genetic variants and contains between 10 and 13 phosphoserine units. It contains 2 cysteine residues and exists as a dimer in milk. Moving on to beta casein. Beta casein consists of 209 amino acids and is approximately 24 kilodaltons. It is the most hydrophobic casein molecule. There are 6 known genetic variants that contain between 0 and 5 phosphoserine units. Similar to alpha S1 casein, beta casein has few secondary structures due to the presence of 35 proline residues. There's also something called gamma casein. Gamma casein is not a primary synthesis product, but is derived by hydrolysis of beta casein by the enzyme plasmin. 3 variants have been identified near the C-terminal end of the beta casein molecule. And finally, the very important kappa casein. Kappa casein consists of 169 amino acids and is approximately 19 kilodaltons, and it contains both glycosylated and phosphorylated residues. It can exist as a dimer up to a decamer with the subunits held together by disulfide linkages. Unlike the other caseins, it is not sensitive to calcium and surrounds the micelles, keeping them intact. It usually contains 1 phosphoserine unit. However, genetic variants containing 2 or 3 have been identified. Further, 9 genetic variants have been identified that demonstrate its diversity. These caseins don't float around individually, they form complex structures called casein micelles. Casein micelles consist of a large portion, approximately 95%, of casein proteins that interact with each other and with calcium. They vary in size from 80 to 1000 nanometers, with an average diameter of 150 nanometers in bovine milk. The micelle sizes of sheep and goat milk are different, and importantly, the caprine or goat micelles are less heat stable than bovine milk micelles. What's inside these micelles? Bovine casein micelles contain water, protein about 94%, and salts about 6%. These salts include calcium, phosphorus, magnesium, and citrate choline, which form something called colloidal calcium phosphate or CCP. They also contain other traces of metals, enzymes like lipases, esterases, and proteases, and milk serum. Casein micelles contain smaller units called submicelles that range from 12 to 15 nanometers in diameter. These submicelles contain approximately 20 to 25 casein molecules and water, about 2 to 5 grams per gram of protein, with some submicelles containing the crucial K-casein. Now, let's explore the other major class, whey proteins. Whey proteins are hydrophobic, globular, highly ordered proteins that contain disulfide linkages. They have well-developed secondary, tertiary, and quaternary structures. However, they have poorer heat stability compared to caseins, and they denature at temperatures greater than 75 degrees Celsius. The 2 principal whey proteins in milk are α-lactalbumin and β-lactoglobulin. They are synthesized in the mammary gland. They constitute approximately 20% and 40% of total whey protein in bovine milk, respectively. Other whey proteins are proteose peptones, immunoglobulins, and serum albumin. Let's look at alpha-lactalbumin first. Alpha-lactalbumin is a spherical, glycosylated, compactly folded calcium metalloprotein. It consists of approximately 142 amino acids and is about 14 kilodaltons. It is synthesized and secreted by the mammary gland. It contains 4 disulfide bonds and 8 cysteine residues and is rich in tryptophan. 3 genetic variants have been identified. And it's a key point that it is the principal protein in human milk. Now for beta-lactoglobulin. Beta-lactoglobulin consists of 178 amino acids with an approximate molecular weight of 18 kilodaltons. It exists in both the monomeric and dimeric form at equilibrium in bovine milk. Its association depends on temperature, pH, protein concentration, and ionic conditions. The hydrophobic dimeric form linked by 1 to 3 disulfide bonds is approximately 36 kilodaltons. There's more to know about beta-lactoglobulin. Higher concentrations of beta-lactoglobulin are present in bovine milk when compared with human milk. It has better heat stability than alpha-lactalbumin due to the presence of 1 free sulfohydryl unit. It contains an open beta-barrel structure, enclosing a hydrophobic cleft and a single 3-turn alpha helix. It binds to several hydrophobic molecules including retinol and fatty acids via the hydrophobic cleft, which in turn stimulates lipase activity. Next we have the protective immunoglobulins. Immunoglobulins are antibodies that are synthesized in response to specific antigens. They are large heterogeneous molecules found in the blood. The main immunoglobulins in milk are IgG, IgG2, IgA and IgM. They provide offspring with protection against pathogenic microorganisms and their toxins, and they also protect the mammary gland itself against infection. Regarding their concentration and structure, approximately 0.7 to 1.0 mg per milliliter is present in bovine milk. The basic structural unit of the immunoglobulins is similar, consisting of two heavy and two light chains joined together by disulfide bonds. IgG is the main immunoglobulin in milk. Another whey protein is bovine serum albumin. Bovine serum albumin consists of 582 amino acids, making it one of the longest proteins we've discussed. It is approximately 66 kilodaltons. It is predominantly composed of alpha helices. It makes up approximately 1% to 5% of total whey protein. It is synthesized in the liver and enters the milk via secretory cells. Then we have proteose peptones. These are derived from the hydrolysis of beta-casein. They are considered whey proteins because they elute in the whey fraction when isolated from milk. They are heat stable. They are acid-soluble proteins. They are mainly responsible for the foaming of skim milk. They inhibit rancidity. And they have an immunological role. Let's talk about a very special protein, lactoferrin. It's a globular glycoprotein, it is about 74 kilodaltons, and it binds to iron as it contains two metal binding sites. Bovine milk contains approximately 20 to 200 milligrams per liter, whereas human milk contains a much higher concentration, around 2 grams per liter, highlighting its importance for infants. There are many other whey proteins with important functions. Other minor whey proteins include growth factors and vitamin-binding proteins for folate, vitamin D, riboflavin, and vitamin B12. Also included are angiogenins and osteopontin. Then we have non-protein nitrogen compounds, like urea, uric acid, creatine, creatinine, and hippuric acid, but these are present only in trace amounts. Now let's bring it all together with a detailed breakdown of the chemical components of bovine milk proteins. First, we have caseins, at 24 to 28 grams per liter. This includes alphas-1 caseins at 12 to 15 grams per liter, with various genetic variants and phosphorylation levels. Alphas-2 caseins at 3 to 4 grams per liter, also with multiple variants. Beta-caseins at 9 to 11 grams per liter, showing many fragments and genetic forms. And kappa-caseins at 2 to 4 grams per liter, with its own set of variants. Second, we have whey proteins Whey, at 5 to 7 grams per liter. This includes β-lactoglobulins at 2 to 4 grams per liter, α-lactalbumins at 0.6 to 1.7 grams per liter, and bovine serum albumin at 0.2 to 0.4 grams per liter. The list continues with more whey proteins. Immunoglobulins are present at 0.5 to 1.8 grams per liter. This category breaks down into IgG immunoglobulins, including IgG1 and IgG2 and their fragments, IgM immunoglobulins, IgA immunoglobulins, including secretory IgA, IgE immunoglobulins, and associated J-chain and free secretory components. Then we have milk fat globule membrane, or MFGM proteins categorized into zone A, B, C, and D proteins. We also have a list of minor proteins, such as serum transferrin, lactoferrin, beta-2 microglobulin, various glycoproteins, ceruloplasmin, a trypsin inhibitor, kininogen, folate-binding protein, and a vitamin B12 binding protein. And finally, a broad category simply listed as enzymes. Moving forward, let's discuss the carbohydrates in milk. There are several carbohydrates in milk: lactose, glucose, galactose, and glycoconjugates, which include oligosaccharides, glycoproteins, and glycoaminoglycans. But the main carbohydrate is undoubtedly lactose. It makes up 4 to 5% of total milk content. Glucose, galactose, and oligosaccharides are present in much smaller amounts, approximately 1 milligram per milliliter. So what exactly is lactose? It's a disaccharide comprised of alpha or beta-D glucose and beta-D galactose that are linked by a beta-1 to 4O glycosidic bond. Its concentration depends on the milk yield and lactation stage of the cow. Generally, the amount of lactose decreases as the lactation stage advances. And lactose exists in 3 different forms: alpha-lactose monohydrate, beta-lactose, and anhydrous alpha-lactose. The form of lactose has practical implications. The beta-lactose form has the greatest solubility and is sweeter than the alpha-lactose forms. Lactose is the major food source for bacteria during the fermentation of milk. The bacteria hydrolyze the lactose into glucose and galactose to produce lactic acid, which inhibits the growth of most other microorganisms, a key principle in making yogurt and cheese. Now let's look at minerals and salts. Milk contains all minerals essential for human nutrition, including potassium, sodium, calcium, magnesium, chloride, and phosphate esters. They make up between 0.7% and 0.8% of total milk content. Sodium, potassium, and chloride exist as free ions, which are readily diffusible. Their concentrations are negatively correlated with lactose, meaning, when lactose is high, these are lower, and vice versa. The balance of minerals is delicate. Concentrations of calcium, magnesium, ionized phosphate, and citrate depend on the casein content in the milk. The concentration of citrate varies depending on the season and diet of the cow. This can affect the soluble calcium content and milk stability because there is a constant equilibrium between the colloidal dispersion and the salts in solution. This equilibrium is crucial for processing. If the colloidal equilibrium is destabilized, the concentration of minerals in milk may affect the processing. For example, it might require the addition of onions. That seems like a possible error. It likely means additives like phosphates or citrates to bind to ionic calcium that would restabilize the caseins against aggregation. Salt is also added as an additive in certain dairy products like cheese. The concentration of calcium in milk is relatively high, which is why milk is considered to be an important dietary source of calcium. What are these salts exactly? The primary salts in milk are phosphates, citrates, chlorides, sulfates, carbonates, and bicarbonates of sodium, potassium, calcium, and magnesium. Since milk contains both organic and inorganic salts, the level of salts is not equivalent to the level of mineral substances. And the level of salts is by no means equivalent to the ash content, which is what remains after burning. Factors influencing salt composition include the species, the breed of species, and the stage of lactation and feed. Finally, let's explore the fascinating world of milk enzymes. In cow's milk, about 20 enzymes have been fully characterized, and another 40 have been demonstrated via their activity. Indigenous milk enzymes are found in or associated with the casein micelles, the milk fat globule membrane, and the milk serum or somatic cells. They may originate from blood, somatic cells, the MFGM, or the cell cytoplasm. Why do we care about these enzymes? They are used as indices of animal health and the thermal history of the milk, telling us if it's been properly pasteurized. Their functions in milk and dairy products are varied. They can result in quality deterioration, they can induce desirable changes, and they may also offer protective effects. Let's look at some enzymes of known or potential technological significance. First is plasmin. It is the predominant indigenous proteinase in milk. It is optimally active at pH around 7.5 and at 37 to the power of circ. Plasmin and its precursor plasminogen originate from the mammal's blood and are predominantly associated with the casein micelle in milk. It plays a role in cheese ripening and affects the stability of casein micelles in UHT milk. Next is lipoprotein, lipase. It has optimal activity at pH 9.2 and 37 to the power of circ, and it's a relatively heat-labile enzyme. It's involved in the initial digestion and absorption of milk lipids in the intestinal tract and flavor development in certain cheeses made from raw milk. However, if it acts unintentionally, it causes lipolysis, leading to the development of hydrolytic rancidity in milk, a major off flavor. A very important one for dairy safety is alkaline phosphatase. It has optimum activity at pH 9.0 to 10.5 and at about 37 to the power of circ. ALP is relatively heat sensitive, and its thermal stability is only slightly higher than that of non-spore-forming pathogenic bacteria. This makes it a perfect indicator of pasteurization efficiency. If the enzyme is inactivated, we know the milk has been heated enough to destroy harmful pathogens. The enzyme's heat stability profile closely follows that necessary for adequate pasteurization. Then we have sulfhydryl oxidase. This enzyme catalyzes the oxidation of thiols and the formation of disulfide bonds in proteins and peptides. There is interest in it because sulfhydryl oxidase-treated UHT milk may have longer flavor stability due to reduced lipid oxidation. Another important enzyme is lactoperoxidase. It is a relatively heat-stable milk enzyme. Heating up to 80 to the power of circ appears required to ensure its thermal inactivation. It acts as an antibacterial agent. It has a pH optimum of about 8.0 and exists primarily in the milk serum. And finally, a quick overview of several other significant milk enzymes. N-acetyl-beta-mine-D-glucosaminidase. Its activity is used as a diagnostic for mastitis test. Catalase is associated with the somatic cell membrane. Xanthine oxidase contains all of the molybdenum in milk. Superoxide dismutase has a protective effect on lipid oxidation. Gamma-glutamyl transferase is involved in the transport of amino acids into the mammary gland. And lactose synthase is the enzyme responsible for the synthesis of lactose itself. And with that, we've reached the end of our detailed exploration. Thank you. See you in the next class. Author